Okay, so we have to continue with our next chapter, which is bioinformatics or proteomics. Okay, so okay. Okay, so here it is. Bioinformatics, crucial part of proteomics. Basically, the name suggests bio means living things. Informatics means all of the databases that you need in order to search for something. So all of the information, if let's say for yourself, you have all of the certificates, the folder that fills all of the profiles of yourself. It's basically a resume of yourself. So it's the same thing for our minute details that we have inside our body and inside all of the living things. Basically, they are stored in the database. Okay, so I'm gonna bring you to hold on. Okay, so this is NCBI. Okay. Okay, let's say you are looking for coronavirus. Okay, and then you can see that there are so many, these refers to all the databases that you need in order to find the information that you're looking for. So they are going to go inside. It will take a while, basically. And time. Okay, and okay, so you can choose if let's say you are looking for the gene. So basically, you know that they are RNA virus. So you can take a look at the gene, but because we are doing proteomics, but it's okay if we spend some time to look at some of the things. And so they basically give you lots of protein information right here. But if let's say you are looking for conserved domains, so some of you might be thinking, what is the meaning of conserved? So how, and some people might think, how are they able to classify them as coronavirus? It's because of the conserved domains, conserved region that they have, which allows them to tell what kind of organism or things or protein basically are the sequence coming from. So conserved region right here. So it says in evolutionary biology, conserved sequences are identical or similar sequences in nucleic acids across species. So for example, if you want to know what kind of things that you have, let's say you have just sequenced something new or novel that has never been sequenced before, at least they would have some percentage of similarities to the available sequence in the databases. For example, I'm going to give you NCBI. So if you would like to look for a sequence that you have on mind. Let's say you have just you did some extraction, you send things for sequencing, and eventually they come out as something. So you can use that information to be blasted against. So there is a blast right here. And let's say it's a protein sequence, so you can choose there as a nucleotide, translate a nucleotide, protein, and you want it to be reverse, translated to nucleotide, protein blast. So you can just choose protein blast and put your sequence right here. Example, let's say I have this information right here. Okay, so all these are the sequences of the okay, so this one is like alignment of something. Okay, so this is domain family. This is an alignment. Okay, let's see. Choose protein. All right, so let's say you have, this is too long, can't read it. Okay, you have this bovine, no, I'm gonna choose, okay, you're gonna choose, I think I've lost it. I used to see that coronavirus somewhere. I can't see it. Okay, it doesn't matter. So you're gonna choose, let's say, SARS coronavirus. Okay, so you choose FASTA. So this one gives you out the sequence that you have. Wow, this is a long one. So in this, this is the FASTA sequence that you can use to copy everything. Okay, let's say you want to do something, let's say you want to do modeling or something. So you can do modeling, but this is basically the information that you have. Okay, but you want to return to the information, the basic information. The name is ORF, or 1AB polyprotein SARS coronavirus. So the locus, the place where it's found, and the length is quite big, which is 7,073 amino acid. It's linear, and this is just the accession number. You can just find it online. And if the key, all of the information are found here. So okay, so basically you can use this information to find, and I mean you can use this website to find the information that you need. Okay. All right, next one. Gonna go to our slide again. Okay. The database is a large collection of data. It has to be very big. If you go to MGI Dunkill, they have a supercomputer to help you store all this information that you need. So basically it's very expensive because you need like a really big place for you to store all of the data. Okay, so and also it requires highly skilled programmers. Okay, and there are also Saccharomyces genome database, which helps you to look for information on Saccharomyces cerevisiae, the one that you have in the kitchen to make bread. Actually, they are really popular in the lab. And there are also other protein databases that you can go and look for, such as UniProt or SwissProt. This is quite popular. I used it during my time, and show you. Okay. Swiss Pride. Swiss Pride. So we're gonna go here, Swiss Pride or Ecstasy. Okay. Okay, so they are basically focusing on coronavirus right now. And if let's say you want to do, you can just click on anything. Let's say you want to do a Swiss model. Okay, so you have, this is the figure that has been recorded previously using the electron microscope. But what we want to do now is, if let's say it has never been modeled, okay, so okay, look at this one. We have a copy like proteases. You can do modeling right here. You can see. Okay. And you need to insert your own protein sequence. I'm gonna find one that is short enough for us to use. Let's say this one right here, which is FASTA. Okay, so I'm gonna just copy it here and paste. Okay, and build a model. Because this is really, while they are building the model, because they are doing calculations, so we can go to another website, which is Protein Blast. While waiting for the Swiss model to do your modeling, you can go to Protein Blast again. If let's say you have forgotten which sequence you have been copying and pasting randomly without knowing wherever it has been taken out from, just go click Blast. This is the one that we have previously from Blast P on NCBI. Okay, so it takes some time to give the result because they are trying to match. Okay, so if let's say you have a sequence that you don't know what the ID is, you can just put in your sequence, and they will come out as percentage of identity as 100%. Similar to transmissible gastroenteritis virus. So basically, if you don't know what's the protein you have, let's say you have just sequenced a new protein from unknown sources or probably novel sources, you can put your information in Blast and you blast it and they will come out as something like this. They give you the percentage of identity. And if let's say it's different, it's going to be, let's say it's novel, so it's not going to be 100% right here, but it's going to be probably some percentage that is similar. So you should expect to see something that is other than 100 or 90%. Okay, so we go back to our modeling. Let's see if it is done. So it's not done yet. Okay, so those are the websites that you can try to play around with. All right, so it has come up with a model like this. And these, okay, you can do rotation. That's the sound of my daughter right there. Okay, so this is the modeling of the protein. So some of you might be wondering, why do we need to know how does the protein looks like? Actually, it is really important for you to, sometimes some people say, oh, crystallization, they do protein crystallization for nothing. Actually, some people don't really understand that the crystallization is so that you can actually know what is the structure of that particular protein you had. And by knowing the structure, you can see where do the if let's say it's an enzyme, so you know where is the active site and where it docks. So okay, so that's the importance of you knowing the active site. And another thing, if let's say you have a protein that is very interesting to be targeted, for example, coronavirus and RNA. So when you have usually the drug that we try to do, it has to target, it has to be in a protein form so that it is easy to target on the model, okay, on the model. So protein and protein, they bind, so the drug will help you out. So that's why sometimes we need to know what is, how does the protein looks like, so that we can create a drug, so that you can find where is the place for us to dock on the specific protein, so that it becomes a perfect drug, or perfect targeting drug. Okay. All right. So we move on to our slide again. Okay. So you can just go try one by one all of these interesting sites online, and when you crawl, there are so many things that you can try. And if let's say they give you out information and all of the things that you need to know in subcellular location and all this. Okay, I can also, and I'm gonna show you one more thing right here. Okay. Taxonomy. Coronavirus. Even coronavirus. Oh, probably I'm gonna do it this way. Coronavirus. Okay, I'm just gonna go choose whichever, whenever. Okay, so you can see the taxonomy. It's not here yet. Probably phylogenetic tree. And sometimes you can go and find what is the relatedness of the thing. Like, let's see. So I should give you the Okay, so find related data. Let's see. 13 words. Let's say just whichever. That's a crystal structure, which basically use X-ray diffraction in order for them to look at the structure. And identify conserved domains. Okay, I think you can just go and find it on your own. So yeah, I think that's at least for today. Okay, so other aspects that will be covered later in the course: protein annotation, identification, edited speech, matching of aspect data. All these, at least I hope I have given you some of the information that you need, and I'm gonna see you next time. Thank you for watching.